Everyone to our online training program. I want to give you today a brief overview of what we plan to do, and hopefully we can have a short discussion at the end of this, hear how you decided to join the program and participate. We really want to get to know you because a big part of this program is working together and not just us delivering these online lectures. So first of all, the goal of this program is to really talk about bioinformatics, but in the context of biological research. So the field of bioinformatics has evolved and today faces the challenge of big and complex data that is readily available, right? We all know about NCBI and other repositories that contain a lot of this data, but getting something useful out of that data is a lot of times a challenge. And so this OmicsLogic program brings together biologists and bioinformaticians to work together and apply analysis methods to next generation sequencing data with an emphasis on the logical connection between the sequencing data, the experimental design, and the meaningful utilization of advanced analytical methods to address biological questions related to basic research, biotechnology applications, and human health. And I think it's very important to understand the logical connection between experiment design, sequencing methodologies, and analytical methods, because at the center of all of these steps, there are people. And these people have to speak a common language. And so that's what the program was focused on, is developing that language that connects the dots between the lab, where the biologists need to know in what ways to improve the data collection and in what ways the data is needed, what more data is needed to answer a research question in a comprehensive and reliable way. They need to understand how the bioinformaticians will use this data to test a hypothesis and maybe train a new machine learning model with this data. And bioinformaticians also have to integrate multiple data sets. So it's important to understand what considerations go into their ability to take data that is normalized, that is ready to be analyzed, and that could be integrated. And at the end of the day, the people that work with this data, that make the decisions based on this data, also need to be aware of that whole process, where the data originated from, how it was analyzed, and how it could be trusted, where to expect some batch effects or overfitting or even lack of representation. And so that's why we want to talk about biology as data science. But when we talk about data science or bioinformatics, it could be uncomfortable or even a little bit uneasy for people that do not have the necessary mathematical, computational, statistical background to utilize these advanced methods. And so what we wanted to do is simplify the tools and the projects enough so that anyone without any coding experience and without any mathematical background that is not ready to maybe invest the time to learn the coding and to learn how to solve complex mathematical problems, still to be able to utilize the methods to interesting projects, the projects that are interesting to you in terms of research and maybe application, and so that we can get that process started, right? So it's an opportunity to kind of come together, focus on specific challenges, and try to do this independently with some hands-on exercises that could accelerate the process. So over the past several years, our team has conducted a number of different programs at university campuses around the world. We have been working extensively with faculty, researchers, and students. And as a result of these different experiences, what we have done is we have developed these online training resources. So essentially, we're talking about 6 online modules. These modules are going to be available throughout this program for all of you to supplement these online meetings. These include an introduction to bioinformatics that talks a little bit about the different ways bioinformatics has addressed specific challenges, the different methodologies, and different exciting projects that we will be covering in this program and that are covered in part on the website. Genomics so genomic data like single nucleotide variation and abundance of mutations in different regions, etc. Transcriptomics, gene and isoform expression, and how gene and isoform expression could be studied using various methods. Epigenetic variation like histone modification and DNA methylation. Metagenomics, which includes the study of amplicon 16S and 18S ribosomal RNA, as well as full 16S metagenomics, and an introduction to machine learning. So as you can see, a lot of these data types are based on next generation sequencing. So that is why the title of our program is bioinformatics, but in the context of next generation sequencing and utilizing different analytical approaches, including machine learning approaches, to the study of this data with a focus on biological discovery. So for the next 3 months, you will have access to all of our online training resources. Sorry, somebody asked here to annotate. So, okay. At the same time, what you will find is that our team will be available to assist you with any questions or technical challenges that you may face. So the way that these materials are structured is something that we will go over. Everybody now has a registration and account on our educational website. And on that account, you and our program managers will be able to see all of the data about your progress. So you and us, we will see how much progress you're making with online resources, with projects, the practical assignments that we have, and we will try and use this data so that we can best assist you to move forward in a project. So first of all, I hope that all of you have an email from Sahil that will get you to this page, to the educational website. And let me switch over from the presentation to the website just to kind of show you how to navigate those resources. So on the so all of you have an email that has your account information that you will find that you will be able to log in to the website. And on the website, you will find different resources here on top. Now, throughout the program, you will be able to access all of our paid materials for free. That includes some of the paid projects and some of the paid courses. So if you're having trouble getting access to any of those, make sure that you are talking to Sahil, which is going to be right here in this chat box, that you will be able to ask for the discount codes or ask for any other clarifications. So now, as you can see my screen, this is the home page. And if you are logged in, you will be able to see your profile right here. Now, please, if you can go into your profile and make sure that all the information there is up to date, and I will show you a little bit what we will be seeing from the interactions throughout this program. If you scroll all the way down, you will see this link under organizations for bioinformatics training. Okay, I see that someone cannot hear anything. So let me know if you have trouble hearing what I'm saying. I can try and speak up louder. Or, like you said, we will have a recorded version of this. So going back to the website, this page is dedicated to our program. So you will see here upcoming events. So right now, this is the event that we are on, and you can see all of the upcoming events and their dates and their time, and some of the things that we will cover on that event. Now, you do not have to register individually to all of these events, but it would be helpful for us to know who is planning to attend so that we can prepare the right materials and send them out to you. Just in case, we will also post information relevant to this to the upcoming session right here on this page. For example, I am just going to ask you to fill out the form that will give us a little bit more information about your educational and research background. So now if you click on this form, you will see that, again, we know that we have some of this information. Just so that the information is consistent for everybody, we want to make sure that you are able to fill out your email, some of your educational background, etc. Now you will see that some of this information will be available on this page that I am showing right here, but you will also be getting emails from Sahil, and please make sure that those emails don't go into your junk folder. You also will be getting various notifications from the different things that you do practically. So just look out for those emails with the t-bio.info domain name. What you will also see here is we have a forum. On the forum, you're welcome to ask any questions or anything that you would like us to discuss. And we will be posting here the recorded sessions for these sessions that we hold online. You can also go to the curriculum tab, and in the curriculum tab, you can see what we are trying to cover in each session in more of a timeline format. And you will see here at the bottom how to reach us all and ask any questions or ask for any materials that were shared. Now, you will also see that you will have the members page, and we hope that you will also find the opportunity to connect so that you can work on projects together or potentially answer any questions in that format as well. So this is a little bit about the page dedicated to the learning program, the OmicsLogic online training program. You will also find courses over here, and these courses are going to be something that you will supplement the materials that we go over through the sessions in person like this. Now, I also want to talk a little bit about your profile. So this is my profile, and you can see here that you will have this section right here with points that shows how much progress you are making. Now, our goal, and we hope for all of you, to get to 10,000 points through the course of the next 3 months. What does the 10,000 points consist of? You will see that under courses, under projects, under points and progress, all of this information is going to be recorded. So you can see that after you complete a course, after you complete or start a course, and after you complete any quiz or any assignment, you will get points, but also you get points for running pipelines. And we'll talk a little bit about running pipelines in the next few minutes. So here, for progress, you can see example of points for passing a course or for running pipelines right here. Okay, so going back to the presentation, I just want to show a little bit how the content is organized. So each one of the online modules is going to be an online learning experience where you will see short sections. So all of these sections have an estimated time to complete, so please take a look at some of those. You'll also be able to download PDF versions of these materials if you want to look at them offline. And what we will see is that a lot of the theoretical materials are supported by practical exercises. The practical exercises will take you from the educational site to our analytical platform called server.t-bio.info. So it's the same platform, but this is the analytical practical component. You will see that each section under the next generation sequencing type of data is organized in a very similar way. You always have the inputs, which are here on top. We have some preloaded genomes on here and different file formats that you can use, and we will provide you with some initial data sets that you can work through. Also at the bottom, you will see that each step of your analytical pipeline will be annotated with informative pop-ups and reference publications that you can read more about the methods that we are using. Now, this is the server.t-bio.info interface, and you should have gotten an email with your account for this as well. So let me quickly switch over. And once you log in, you will be able to see that here you also have your profile with your email address and with some information about you. So after you receive that email, you can click on login, and here you will see your email is your username, and this is your password, and you can click on remember me so that you will see your profile. Now here, under each section, are you seeing, yeah, just making sure that you can see, okay, can you see my screen, Justin? Okay. Okay, so here you will see that each section is divided into these subsections, and you have a number of applications for next generation sequencing data. So we will focus on some of these sections right here, as well as some of the sections under data mining, utilities, and some of the sections under data association we will touch upon. Now you will see here as well, there's a chat box, again with the heel, and any trouble you have logging in or accessing anything, you can always put it right here, and we will hope to address any questions in a very timely manner. Like I said, each section works in a very similar way, so we will talk more about this next time, and you can also look at some videos that we will send you after this session on how to utilize the platform. But essentially, what you will do every time is you will select some type of a data format, for example, an organism, so a genome that you would like to align the sequence to, and here you'll be able to upload files. Now, in this program, we will provide you with data sets that you will use either with SVO links directly to FASTQ files that are already uploaded on the server, or whole collections of data that will come in a single file. And we'll go over how to technically do this in the future. But for example, if I have a file that is called edu.t-bio.info, now I will be able to use this file to build my pipeline. If somebody's sound is gone, so I can. Thank you. Okay, so now you can see that the platform recognizes that the files have been uploaded. If I don't need to use groups, now I can build my pipeline. So here you will have darker color buttons that represent the selection that you can make with the type of data that you uploaded. And then once you select any kind of a method, you'll be able to read about that method and oftentimes go to the publication relevance, and you can see the connection between the steps that you're making. And all of these are also color-coded, helping you to understand the logic of the inputs, the outputs, and what to expect when you have completed your pipeline. So we're not going to really do anything with this right this second, and we will try to move on into the practical aspects of the platform after we complete our next session. But once you completed your pipeline, you save it, and you can run your pipeline. Do not forget to run your pipeline, because otherwise it will be just prepared. So you can see under my pipeline, this one is prepared. If I go back into the pipeline, I can see what I've prepared, and I can run it. And if I don't run it, it will just stay prepared. We will also be using some demo pipelines that we'll talk a little bit more in our next session. Okay, so the platform is really built to allow this kind of an interactive way to launch algorithms and deploy them on large data without having to rely on the computational resources that you have available in front of you. So it does not use any of your hard drive. It does not use any of your RAM on the computer. This is all cloud-based, and all of the files are already pre-uploaded. And once we are ready to work on an independent project, we'll help you get those files in there so that you can practice and work with them. Now, the other aspect is that we will learn how to look at data and understand what this data means. So we will try and link the data that we got as an output from the pipelines with how to analyze and get some meaning, some information that we can answer questions about some biological processes. We will try to link that primarily with phenotypes. So how to connect next generation sequence data with some observed phenotype at different levels. The trainers in the program will have the opportunity to address different aspects of the projects and the topics that we are going to be covering. So I also already mentioned Sahil. Sahil is going to be primarily responsible for technical support and making sure that you have all of the files, all of the recordings, your account information, and anything else that is needed. Justin is heading up our educational materials development, and he is going to be talking about some of the biological backgrounds of the projects that we are studying, and he will help answer any questions on the biology perspective for these projects. Julia is currently a PhD student that is going to help understand some of the methods that we will utilize to analyze and process next generation sequencing. And Mohit is going to be assisting in India primarily with the coordination there, but also he is a computational biologist himself, and so possibly he will help address some of the computational and machine learning methods that we will discuss throughout this program. Now, the program is not only our Pine Biotech team, but we also rely on consultants and advisors from our partners at the Tauber Bioinformatics Research Center, which is a research center based in Israel at University of Haifa. And the people at that center are the primary developers of the methods and the platform itself. And they will help us address any technical challenges or sometimes explain the complex bioinformatics approaches that we will be utilizing. Now, the idea of the program is that we will look at several publications. And these publications typically include their experimental data. So we will rely on publications that have next generation sequencing data, and it will utilize some methods. And these methods have been published and digested and applied to address a specific challenge. So we will try to link those two by reading that publication, and we will use that publication to also ask you several questions to make sure that we're all on the same page and we understand correctly why specific methods were used and why the data set, how the data set was analyzed and what were the results and how to understand those results. But as a result of this process, we will see that there's a connection between data and methods. And by trying other data analysis methods and other data that is similar to what we studied, we will be able to develop a novel and independent project. So a project that could be called independently yours, and you are welcome to work in teams or together, or even just to observe and learn how these similar projects were already developed. So we will provide you with a starting point with many different types of projects that we have prepared and you can start with. It's up to you which one you pick, and we will kind of spend a little bit more time on several of these. But just to give you an idea of what types of projects we have prepared for this program, we have some analysis, epigenomic analysis of human T-cell differentiation in asthma patients, modeling precision treatment of breast cancer, viral mutation landscape study using ultra-deep seq sequencing of polio, agriculture and environmental programs, projects related to toxic resistance in pine pathogens, changing immune response in cancer with tumor-associated macrophages. We'll look at microbiome biomarkers from the American Gut Microbiome Project, whole transcriptome profiling of cancer tumors in mouse PDX models, deregulation of subcellular network in cancer patients based on Cancer Atlas Genome, TCGA, insect cell line biologics production, so SF9 cell lines and raptovirus contamination published by the FDA, crop drought stress and resistance in potato, different crops, potato crops, complex gene regulation in neurodegenerative diseases. So this is going to talk about the Angelman syndrome. And by the way, for those of you that are interested in neurodegenerative diseases, we are looking at an upcoming conference in Louisiana, where at the annual bioinformatics conference, we will have an expert on Angelman syndrome, Dr. Kafzan from University of Haifa, and Julia Panov, who is going to be presenting, doing a hands-on workshop. If you're willing to travel to Baton Rouge, to LSU, and participate in that hands-on project, you're invited and you can attend that for free. And also join the session that Dr. Kafsan will lead on Angelman syndrome on April 6th. So if you're interested, please let us know and we'll help coordinate the times. So please, you can reach out to Sahil about that. We'll talk about Ebola, deadly mutations, study of the West African epidemic in 2013, 2016. Obviously now it's recurring, so it's important to understand how those Ebola strains evolved. And you will be able to develop your own project as well. So if you are interested in a specific data set, we are not going to jump to that right at the beginning because we believe that preparing for the project and planning your analysis is critical for the success. So all of these projects are based on publications, like I said. And when we look at them, we extract the data and we help build those connections between how the data was prepared, where it was taken from, how to analyze it, and then what conclusions could be made. And we'll try quite a number of different methods to see the ways that those could be used effectively, and also understand why some approaches are not going to be very effective. Now, in that, those that have completed our program have presented individual posters or team posters on the project that they have done. And so all of you, I know the primary background are faculty and PhD postdocs. So, this could be a department seminar. This could be participation in some kind of a conference. This could be used in application to your own data. So if that is an outcome that is interesting to you, then we are interested to help you get there as much as possible, but it will require some coordination. So the sooner you reach out to us about it, the better we can help you in the long run. As I mentioned, we will be monitoring a lot of the activity on the educational website, on the server platform where you will run the analysis, to try and collect some data, also using quizzes and surveys to try and collect data on participation. We are really trying to develop this program into a very high quality program that we can speak about our achievements and not just the process. And so we're trying to make sure that everybody reaches a certain level of competency when we say that they have successfully completed this program. We will be trying to reach out to you individually or through our representatives to make sure that everybody is making good progress in a timely way. Obviously, if you're interested at the end, you will be able to get a certificate of completion on this 3-month online training program. If you are a faculty member and you are considering to utilize some of these resources in your education, in the training that you conduct at your university, please let us know. We have some explanation about how this whole program works on this link on this website. So you can look at this page. Sorry. So I'll just show you real quick how you can learn more about these resources. And also, if you are interested in that, then you can definitely reach out to Justin. Justin will be able to explain how the program works and how we are using this program in several universities today, including Georgetown University, University of Grambling State University, LSU, and some of the Louisiana universities, University of Haifa, and others. So you can read a little bit more about this, and basically what I've told you today about the online resources, about these sessions and hackathons that we conduct, and some of the types of workshops that we have prepared, all of this comes in a package that you could implement at your university if you were willing and interested to do so with our support, of course. What are we going to cover in this program? So in this program, we will review how the world of next generation sequencing has changed the way we can study molecular and cell processes from the way data is stored in the DNA to the way that it is controlled in epigenetic, using different epigenetic variation, and how it gets transcribed, and we can study that with gene expression. And then we can also do a lot of things with the available data sets that we have online in different repositories. And so we can mine those big data sets to try and understand some aspect of that biology and then learn how to validate those findings using other data sets. And we'll talk a little bit more about how to structure a bioinformatics analysis project and how to present it and how to talk about these different analysis methods that we are going to utilize to make sure that they were fine-tuned to really address the problem that we are trying to solve. So now what I want to do is just talk a little bit about previous programs that we ran and how this might be useful to you, to different types of people that are participating in this program. So in the past, when we conducted these programs, we saw that the majority of the people fell aside from the program if they attended most of the online sessions and not watched them independently with recordings. Because in the online session, you can ask questions, we can have a discussion, we can dive deeper into some aspect of what we're talking about. And all of that is more relevant if you let us know how it's interesting to you or what is it that you find most challenging. So having that dialogue is very important. Now, obviously, during the session, we can do that, but the sessions are only 1 hour long. So having that information in advance is going to be very helpful to us. I also think that most of the people that we worked with in the past benefited from the program if they had some free time to go through the online materials. Now, these materials do not require too much time. Some of them could take an hour and a half. Some of them could take a little bit longer to complete. But if you complete some of them and you have some technical challenges, then we would be more prepared to address those in the sessions. If you don't do any of the online materials and you just jump to the practical things we do in the online sessions, you might lose out on some of that information. So that's a little bit about the program, about our team, about what we're trying to do. And now we have some time, and I would like to start by letting Sahil, who's going to be the primary coordinator for the program, say a few words about his background and kind of how he is going to be involved in this program. And then after Sahil, Justin will say a couple of words about the educational aspect and how, as biologists and as researchers, utilizing this data, we can get sufficient insight from the tools and from the projects that we will be going through. And also give an opportunity to all of you to introduce yourselves and say a few words about why you joined and what do you expect to get out of this program. So, Sahil, do you want to take it over?
Yeah, sure. Thank you, Leah. Hello, everyone, and welcome again to the program. My name is Sahil Sethi, and I have a background into bioinformatics. I have done my bachelor's in bioinformatics from India and my master's in biomedical informatics from University of Nebraska at Omaha. And I've been working with Pine for an year now. And I mean, it's great working with Pine, and I've been interacting quite a lot with the participants in the previous programs. I am a liaison between the participant and between the education coordinator and Ilya, definitely. So if there is any problem, any questions, any issues that come along, please feel free to reach out to me. I try to be pretty responsive when it comes to replying to the questions. And the only one thing that I would like to request all the participants is like, be proactive, once the session is done. So, I mean, just take out like couple of hours from your daily schedule so to as to go over like what was covered in the previous lecture. Um, Try out some pipelines, like do some hands-on and go over the online modules. So that would give you a clear understanding about what happened in the previous lecture and then what's gonna come in the next lecture. So that would help you in maintaining the pace of the program, and then that's when you're gonna benefit most out of it. And at the end, definitely when there's a chance to work on a project, so you'll have a better idea to work on your own project. And I would like to wish good luck to all the participants and hope you guys enjoy it. Thank you.
Justin?
Can you hear me?
Yeah.
Okay. So this is Justin. I am a neurobiologist in training. I got my master's in applied physiology, and I got my PhD from Brandeis in molecular and cellular biology. And there, I specialized on stem cell differentiation in the nervous system during development. I did my postdoc at the National Institutes of Health. I've been in academics for the past approximately 5 years. And more recently, I worked for a video education company called Jove, which a lot of you might have heard of. So, varied exposure and experience as far as my role at Pine. As Ely mentioned previously, I think, general biology related questions or biochemistry are related, and how those fields, what we offer in those fields as far as video support and general questions about the fields themselves. And I think more relevant, I can be of use with respect to mapping curriculums and integrating the content into your current systems. And I think that's one of the limitations as faculty or postdocs. Time is limited, you get into your groove, things are working, but I think we also know that mixing it up, trying new things, trying new approaches to reach the students is very important. So I'd like to help all of you make that transition using our systems, make it as painless as possible. And also, as Yulia mentioned, I think one of the important things with a support network and using a new bioinformatics tool also requires ongoing support, right? You can't just start using it and that's it, right? It'll drop off after a couple of weeks. You'll be frustrated. Why did I do this? So, absolutely, short term, long term, I would very much like to help you with that process, and I find it very satisfying, to help fellow academics and scientists, excel using our material.
Thank you, Justin. And we do have 10 minutes left here, so I do want to invite any of you, if you have any questions or you would just like to simply introduce yourself. I think one of the major things that we've seen in the past is that people get to know each other, they're able to collaborate and work on some of these practical assignments and get feedback. Especially if you're in the same location. I know that we have people from several locations, and I think there's a lot of benefit to conducting this kind of a program online so that you can do it from the comfort of your home or your office. But at the same time, that interpersonal connection plays a critical role in the success of the overall program. So if any one of you would like to say a couple of words about where you are now and maybe your position at your university, and if you have any questions, we'd be happy to answer those as well.
Hi, this is Akhil here. Can you hear me?
Yeah.
Yeah.
Sure. So I'm Akhil Rajput. I did my PhD from Weizmann Institute of Science, Israel, and in the field of biochemistry and molecular biology. Worked on program cell death, and then moved to San Diego in an institute by the name of Sanford Burnham. There did research on ubiquitin ligases. But that is when I realized that lots of biochemistry and molecular biology understanding as far as pathways are concerned, which I was gaining, it has to be integrated by employing informatics. And there comes the field of bioinformatics. So made a transition to genomics-based understanding, and that's where I thought that probably taking this course will really help me to understand the basics and implementing that to the clinical sciences. Ultimately coming up with some solutions which can help the patients on the other sides. So that's the motivation, and I'm really looking forward to this fantastic course. And if there are some local people over here in San Diego, I'll be happy to join with them. Otherwise, even building a team and group online, will be a good way to go. So I'm really excited about this journey for 3 months. Thank you.
Thank you very much. Anyone else?
Hi, I'm Laura Schrader at Tulane University. Can you hear me?
Yes.
I am an associate professor in cell molecular biology, and I'm a neuroscientist. I've been in neuroscience for probably about 30 years now, and I would just like to learn more about bioinformatics, and we have some RNA-seq data that I would like to understand how to analyze. And I'm looking forward to the course.
Great. Thank you very much, Lauren. Nice to meet you.
Hello.
Yes.
I'm Nathan. I graduated with recently in 2017 with a degree in biology. I've just had a couple of internships, like a 10-week internship in a genetics lab, a wet lab, and a 10-week internship in bioinformatics. So I'm relatively new, but I'm very interested, and I hope I learn a lot through this program.
Thank you very much. Nice to meet you. Okay, anyone else would like to maybe give us a few words?
Hi, can you hear me?
Yes.
My name is Tito Dokumu. I had my full degree in biochemistry and MSc and PhD in pharmacology. And my research work has mostly been on genomics and infectious diseases. I'm on a postdoc at Southern University, Baton Rouge. So I want to learn a lot more on bioinformatics, which is mostly what my research is based on. Thank you.
Okay, thank you very much. Nice to meet you. So if you are speaking, please unmute yourself because we can't hear you.
Hello.
Hello.
Can you hear me? This is Sagar from Louisiana State University.
Yep.
Yep. I'm a PhD student at immunology program. So I'm finishing up my PhD this year and looking for the next position. And I would like to extend my horizon to the bioinformatics, mostly the analysis of big data, genomic data, so that I could be better prepared for the next position. And I'm really excited about this program. Thank you.
Hello.
Yes.
Can you hear me?
Yes.
I'm Siva Veliveli. I did my PhD in plant science and currently working as a research scientist at Donald Danforth Cancer Center in St. Louis. So my PhD research was focused on plant beneficial microbe interactions. I'm currently working on antimicrobial peptides, and I'm looking forward to learn more about RNA-seq through this course. Thank you.
Hello. Hello, can you hear me?
Hello. Yeah.
Yeah.
I'm Chitto Goto from MIT University, Kolkata, India. Hello.
Yeah, we can hear you.
Yeah. Okay. So I have done my PhD in bioinformatics, and my master's was in biotechnology. So my interest is NGS data analysis, and basically I'm interested in gene co-expression network analysis. So I'll try my best to learn this course. Thank you.
Ayan, I see that maybe are you trying to also say something, but your microphone is muted? Yes, your microphone is muted, so we cannot hear you.
Yeah. So I'm just introducing myself. I am Dr. Ayun Rai Chaudhary from Amity University, Kolkata. I completed my PhD in biochemistry from University of Calcutta, India, and then I completed my postdoc from University of Pennsylvania, USA. And at present, I returned India and working at Amity University Biotechnology Department. So I'm very much interested to understand biotechnological aspects, all these next generation sequencing and so on. So I'm very much interested in this. Thank you.
Very much, and nice to meet you.
Thank you.
Thank you.
Okay, anyone else would like to just briefly introduce yourself, and then we will wrap it up. Okay, well, thank you very much, everyone, for sharing with us. And we will also, like I mentioned, send out the survey. And please let us know as much as you would like us to know about you and your educational background. As you can see, there's quite a variety of interests here, people from different types of biological problems, agriculture, neurodevelopment, health related issues. So we will try to make sure that everybody gets some background information about the different sequencing methods, the different bioinformatics methods, an idea on project planning in consideration of those sequencing, experiment design, etc. factors. And then we will try to give you some initial materials that you can either try, or as it has been mentioned, if you are coming with your own data or with your own project that you have identified, we would be happy to help you get that started as well. So again, you can reach out to us directly, or you can use the survey to let us know about those types of interests. Now, in conclusion, I would just like to say that please make sure that you have your account set up, that your password is working, that you've explored some of the technical things around the platform and the way that the materials are structured. And if you have any trouble with your password, with your email, with something not opening, let's resolve that as soon as possible so that the next session we can dive into the subject matter and not spend any more time on those technical issues. At the same time, I just want to say that we will have, again, this upcoming conference in Louisiana, the annual Bioinformatics Conference in Baton Rouge at the LSU Center for Computation and Technology. We will have a hands-on workshop. If you are in the Baton Rouge area in Louisiana in general, you're welcome to join that. That's going to be a free workshop. It's going to be focused on epigenetic and gene expression data, epigenomic and gene expression data from Engelmann syndrome. And then during the 5th and the 6th, so on the 5th in the morning, we will have that hands-on workshop. You're welcome to participate. We also will have an offline connection. So this is not listed in our schedule, but if you're available during that time, if you would like to join, please join us. We will have Julia speaking about her research and the lab of Dr. Kafzan. Then on Saturday, on April 6th, we will have, and we will send you all of this information by email as well. But on Saturday, on April 6th, we will have several presentations. One of them will be on different mutation patterns that can define adaptation, viral adaptation to host, and we will hold also a seminar at the vet school there at LSU. You will also see that on April 1st and 2nd, we will be in Georgetown in the Washington, D.C. area. So April 1st, we will have a workshop on Ebola, a hands-on workshop on Ebola. This is through the Systems Medicine program at Georgetown University. And then on the 2nd, we will have a seminar on TCGA data and multi-omics integration. So we will hope to make those sessions available as well. If you're interested, you're welcome to join there as well. Any events that we hold, we will let you know. And if you would like to register and attend those, please communicate with us so that we can set you up for those events. Finally, I just want to say that we're very excited about this program. We're hoping that you will get a lot out of this. We will get to work together on some of these data sets, and hopefully the 3 months will be spent in the thank you for joining. Our session is over now, and we will send you a recorded version and a follow-up email. And if you'd like to schedule any kind of conversation with any of our experts here, Justin, Sahil, and Mohit, and Pratim also in around Kolkata area, please let us know. Thank you, and good luck with the program.